Namaskar. Welcome back to the lecture series. In this session, we are going to see the immune response against viral and bacterial diseases. Many human illnesses are caused by infection with either pathogenic bacteria or viruses which are disease causing. To cause disease, these microorganisms must gain access to the body. Bacteria reproduce by splitting into two, while the viruses replicate themselves inside host cells. Many bacterial infections can be treated successfully with antibiotics, but these are useless against viral infections. Bacteria are tiny single-cell organisms. They are among the most successful life forms on the planet and range in habitat from ice slopes to deserts. Bacteria can be beneficial. For instance, gut bacteria help us to digest the food, but some are responsible for a range of infections. These disease-causing varieties are called pathogenic bacteria. Many bacterial infections can be treated successfully with appropriate antibiotics, although antibiotic resistant strains are beginning to emerge. Immunization is available to prevent many important bacterial diseases. A virus is an even smaller microorganism that can only reproduce inside a host living cell. It is very difficult to kill a virus. That is why some of the most serious communicable diseases known to medical science are viral in origin. Let's see how the bacteria and viruses make their entry into the body. To cause disease, pathogenic bacteria must gain access into the body. The range of access routes for bacteria includes cuts, contaminated food and water, close contact with an infected person, contact with the feces of an infected person, breathing in the exhaled droplets when an infected person coughs or sneezes, indirectly by touching contaminated surfaces such as taps, toilet handles, toys and nappies. Viruses are spread from one person to another by coughs, sneezes, vomits, bites from infected animals or insects, exposure to infected bodily fluids through activities such as sexual intercourse or sharing hypodermic needles. Forgetting to wash your hands after handling pets and animals is another way for germs to be taken in by mouth. Virus types. Let's see what kind of virus types are present. A virus is a minuscule pocket of protein that contains genetic material. If you place a virus next to a bacterium, the virus would be drafted. For example, a poliovirus is around 50 times smaller than a streptococci bacterium, which itself is only 0.003 mm long. Viruses can be described as either RNA or DNA viruses according to which type of nucleic acid forms their encore. The four main types of viruses include icosahedral. The outer shell capsid is made from 20 flat sides, which gives a spherical shape. Most viruses are icosahedral. Helical shape. The capsid is shaped like a rod. Enveloped. The capsid is encased in a baggy membrane which can change shape but often appear spherical. Complex. The genetic material is coated but without a capsid. Let's see the body's response to a viral infection. Viruses pose a considerable challenge to a body's immune response system because they hide inside cells. This makes it difficult for antibodies to reach them. Some special immune system cells called T lymphocytes can recognize and kill cells containing viruses since the surface of the infected cells is changed when the virus begins to multiply. Many viruses, when released from infected cells, will be effectively knocked out by antibodies that have been produced in response to infection or previous immunization. How can we cure a viral infection? Just let's have a look into that. Antibiotics are useless against a viral infections. This is because viruses are so simple that they use their host cells to perform their activities for them. So, antiviral drugs work differently to antibiotics by interfering with the viral enzymes instead. Antiviral drugs are currently only effective against a few viral diseases such as influenza, herpes, hepatitis B and C, and HIV. But research is still ongoing. A naturally occurring protein called interferon, which the body produces to help fight the viral infections, can now be produced in the laboratory and is used to treat the hepatitis C infections. Next, immunization against viral infection is not always possible. It is possible to vaccinate against many serious viral infections such as measles, mumps, hepatitis A and hepatitis B. An aggressive worldwide vaccination campaign headed by the World Health Organization WHO managed to wipe out smallpox. However, some viruses such as those that cause the common cold are capable of mutating from one person to the next. This is how an infection with essentially the same virus can keep dotting the immune system, which is very dangerous. Vaccination for these kinds of viruses is very difficult because the viruses have already changed their format from the time vaccines are developed. Let us see how the immune responses work to the viruses via cytotoxic cells. When a virus infects a person, a host, it invades the cells of its host in order to survive and replicate. Once inside, the cells of an immune system cannot see the virus and therefore do not know that the host cell is infected. To overcome this, cells employ a system that allows them to show other cells what is inside them. They use molecules called class 1 major histocompatibility complex proteins, also called as MH3 class 1 for short, to display pieces of the protein from inside the cell upon the cell surface. If the cell is infected with a virus, these pieces of peptides will include fragments of the protein made by the virus. A special cell of an immune system called a T cell circulates looking for the infections. One type of T cell is called cytotoxic T cell because it kills cells that are infected with viruses with toxic mediators. Cytotoxic T cells have specialized proteins on their surface that help them to recognize virally infected cells. These proteins are called T cell receptors, also called as TCRs in short. Each cytotoxic T cells has a TCR that can specifically recognize a particular antigenic peptide bound to an MHC molecule. If the T cell receptor detects a peptide from a virus, it warns its T cells of an infection. The T cell releases cytotoxic factors to kill the infected cell and therefore prevent survival of the invading virus. This particular mechanism is represented in the figure 1. Viruses are highly adaptable and have developed ways to avoid detection by T cells. Some viruses stop MHC molecules from getting to the cell surface to display viral peptides. If this happens, the T cell doesn't know there is a virus inside the infected cell. However, another immune system specializes in killing cells that have a reduced number of MHC class I molecules on their surface. This cell is a natural killer cell or NK cell for short. When the NK cell finds a cell displaying fewer than the normal MHC molecules, it releases toxic substances in a similar way to cytotoxic T cells, which kill the virally infected cell. Cytotoxic cells are armed with preformed mediators. Cytotoxic factors are stored inside the compartments called the granules in both cytotoxic T cells and NK cells until contact with an infected cell triggers their release. One of these mediators is perforin, a protein that can make pores in the cell membrane. This particular perforin protein allows entry of other factors into a target cell to facilitate destruction of the cell. Enzymes called granzymes are also stored in and released from the granules. Granzymes enter the target cells through the holes made by the perforin. Once inside the target cell, they initiate a process known as programmed cell death or apoptosis, causing the target cell to die. Another release cytotoxic factors is granulizing, which directly attacks the outer membrane of the target cell, destroying it by lysis. Cytotoxic cells also newly synthesize and release other proteins called cytokines after making contact with infected cells. Cytokines include interferon and tumor necrosis factors and transfer a signal from the T cell to the infected or other neighboring cells to enhance the killing mechanisms. Let's see how this happens via interferons. Virally infected cells produce and release small proteins called interferons, which play a role in immune protection against viruses. Interferons prevent replication of viruses by directly interfering with their ability to replicate within an infected cell. They also act as signaling molecules that allow infected cell to warn nearby cells of a viral presence. This signal makes neighboring cells increase the number of major histocompatibility class I molecules upon their surfaces, so that the T cells surviving the area can identify and eliminate the viral infection as described above. Let's see how this mechanism works out via antibodies. Viruses can also be removed from the body by antibodies before they get the chance to infect a cell. Antibodies are proteins that specifically recognize invading pathogens and bind or stick to them. This binding serves many purposes in the eradication of the viruses as follows. Firstly, the antibodies neutralize the virus, meaning that it is no longer capable of infecting the host cell. Secondly, many antibodies can work together, causing viral particles to stick together in a process called agglutination. Agglutinated viruses make an easier target for immune cells than single viral particles. A third mechanism used by antibodies to eradicate viruses is the activation of phagocytes. A virus-borne antibody binds to a receptor called Fc receptors on the surface of the phagocytic cells and triggers a mechanism known as phagocytosis, by which the cell engulfs and destroys the virus. Finally, antibodies can also activate the complement system, which opsonizes and promotes phagocytosis of the virus. Complement can also damage the envelope, the phospholipid bilayer that is present on some types of viruses. Well, let's come to the bacteria types. Bacteria that cause diseases are broadly classified according to their shape. The four main groups are: the first group are the bacilli, shape 1. They are like rod with a length of around 0.03 mm. Illnesses such as typhoid and cystitis are caused by this bacilli strains. Secondly, it is the coccus shaped. They are like spear with a diameter of around 0.001 mm. Depending on the sort, coccus bacteria group themselves into a wide range of ways, such as in pairs, long lines, or tight clusters. Examples include staphylococci, which cause a host of infections including boils, and gonococci, which cause the sexually transmissible infection gonorrhea. Then spirochetes, as the name suggests, these bacteria are shaped like tiny spirals. Spirochetes bacteria are responsible for a range of diseases including sexually transmissible infection, syphilis. Then thirdly, the Vibrio, shaped like coma. The tropical disease cholera, characterized by severe diarrhea and dehydration, is caused by the Vibrio bacteria. Now let's see the characteristics of this bacteria. Most bacteria, apart from the coccus variety, move around with the aid of small lashing tails called the flagella, or by whipping their bodies from side to side. Under the right condition, a bacterium reproduces by dividing in two. Each daughter cells then divides in two and so on, so that a single bacterium can bloom into a population of some 500,000 or more within just 8 hours. If the environmental conditions don't suit the bacterium, some varieties morph into a dormant state. They develop a tough outer coating and await the appropriate change of conditions. These hibernating bacteria are called spores. Spores are harder to kill than active bacteria because of their outer coating. Let's see how the bacterial infection can be cured. Curing a bacterial infection. The body reacts to disease causing bacteria by increasing local blood flow, the inflammation, and sending in cells from the immune system to the attack and destroy the bacteria. Antibodies produced by the immune system attach to the bacteria and help in their destruction. They may also inactivate toxins produced by the particular pathogen, for example, tetanus and diphtheria. Serious infections can be treated with antibiotics, which work by disrupting the bacterium's metabolic processes. Although antibiotic resistant strains are starting to emerge, immunization is available to prevent many important bacterial diseases such as Haemophilus influenza type B, called the HIB, tetanus and whooping cough. Let's see how the immune responses are there towards bacteria. If you see via complement mediated lysis, when bacteria such as Neisseria meningitidis invade the body, they are attacked by immune proteins called complement proteins. Complement proteins assist in bacterial killing via three pathways: the classical complement pathway, the alternate complement pathway, or the lectin pathway. The first steps of a classical complement pathway require the binding of antibodies to the surface of the target bacterium. The antibodies then become target for one particular complement protein complex known as C1. C1 binds to the tail known as Fc region of the antibody. Once bound, C1 initiates a cascade of cleavage and reforming of complement complexes that ends in the binding of several complement proteins to the surface of the bacterium in the form of a membrane attack complex called the MAC complex. This particular mechanism is presented in the figure 1, or it can also generate opsonins that label a bacterium for destruction. MAC can insert into the cell membrane of gram-negative but not gram-positive bacteria. Therefore, it produces pores that allow the entry of the membrane, damaging the molecule such as lysozyme, and makes the bacterium susceptible to osmolytic lysis. When you see the other pathway, which is called the alternate complement pathway, this particular pathway does not require antibody to initiate the lysis of the bacteria. In this pathway, complement proteins form a complex known as C3, directly bind to the bacteria and activate downstream components in the complement cascade, once again ending in the formation of MAC that causes lysis of the bacterium. If you see the other lectin pathway, in this, during the lectin pathway, the mannan binding lectin, which is called as MBL, binds to the proteins containing mannose residues that are found in some types of bacteria such as Salmonella species. Once bound, MBL forms a complex with an enzyme called MBL activated serine protease. In short, it is called as MASP. In this form, this enzyme activates C3 convertase by cleaving C2 and C4 complement components that participates in forming MAC. Next, if you see how this pathway goes via phagocytosis. In bacteria, the bacteria may also be killed by phagocytes. Immune proteins like acute phase proteins like complement and antibodies bind to a surface of bacteria by a process called opsonization. Opsonized bacteria are therefore coated with molecules that phagocyte cells recognize and respond to. Activated phagocytes engulf and destroy opsonized bacteria by a process called phagocytosis. Complement C3b is particularly important opsonization protein for controlling the bacterial infection by this mechanism. Opsonization allows killing of the gram-positive bacteria, that is example Staphylococcus species, that are resistant to killing by MAC. After bacteria are ingested by phagocytosis as illustrated in figure 2, they are killed by various processes that occur inside the cell and broken into small fragments by enzymes. Phagocytes present the fragments on their surface via class II major histocompatibility, that is MHC class II molecules. Circulating helper T cells recognize these bacterial fragments and begin to produce proteins called cytokines. Two major groups of helper T cells are known as Th1 and Th2 cells. These cell types differ in the types of cytokine they secrete. Th1 cells predominantly produce interferon G called the IFN-G, which promotes cell-mediated immune mechanisms. The Th2 cells produce mostly interleukin-4 called the IL-4, which promotes humoral immunity by activating B cells. B cells make antibodies that stick to extracellular bacteria and prevent their growth and survival. Well, now let us see how it happens via cell-mediated immunity. Some bacteria engulf during phagocytosis, avoid the killing mechanisms of the phagocyte to survive inside the cells. Macrophages are a common target for intracellular bacteria, example Salmonella species, that live inside the cell compartments. These bacteria cannot be detected by complement or by antibody, but instead are eliminated using a cell-mediated response. Infected macrophages present bacterial peptides on the cell surface using major histocompatibility class II molecules. This mechanism is called antigen presentation. A helper T cell surveys MHC class II molecules and its T cell receptors, the TCRs, to observe the peptides they hold. If a bacteria peptide is presented, the Th1 cell releases interferon G. This cytokine stimulates killing mechanism such as production of lysozyme inside the infected macrophage to digest and destroy the invading bacterium. Interferon G also increases antigen presentation by cells, making the bacterium more visible to the immune system and more prone to attack, which is illustrated in the figure 3. Therefore, we conclude this session. We have seen how the immune response against viral and bacterial diseases is being carried out in the biological pathways. I hope you enjoy the session and continue listening to the lecture series. Thank you.